Chemiosmosis by Kayla Raddick and Amelia Webb. In 1961, Peter Mitchell proposed his chemiosmotic hypothesis. His hypothesis suggested that ATP was created via an electrochemical gradient in the inner membrane of mitochondria and chloroplast. Enough evidence has been compiled that his hypothesis is now considered a theory, and in 1978, Mitchell was awarded the Nobel Prize in Chemistry. Chemiosmosis is the movement of ions across a selectively permeable membrane down their electrochemical gradient. This occurs in the second step of oxidative phosphorylation. Unlike other types of diffusion, ionic diffusion requires a carrier to help them move across the membrane, more specifically the use of ATP synthase through facilitated diffusion. So let's take a closer look. When we digest food, the sugars enter the citric acid cycle and produce NADH and FADH2 that will then put hydrogen ions into the electron transport chain, a lot of hydrogen ions. Remember, one glucose molecule sets 2 citric acid cycles into motion, creating a total of 6 NADH and 2 FADH molecules. This buildup of hydrogen ions outside of the membrane creates an electrochemical gradient. The ATP synthase enzyme has 4 major components: the stator that anchors the complex in the membrane, the transmembrane proton channel that allows protons to move down the gradient, and the rotor and rod that spin together to open the catalytic component known as the catalytic knob. 2 hydrogen ions enter the complex. 1 hydrogen ion will pass through the complex and enter the matrix space. The other attaches to the rotor and causes the rotor to turn. Once 3 hydrogen ions have attached to the rotor, enough energy is stored to twist the rod and turn the catalytic knob. This allows adenosine diphosphate to bind with a phosphate inside the catalytic knob to form adenosine triphosphate. This ATP will go on to provide energy for all cellular functions within the organism. This process is repeated until equilibrium for the hydrogen ions is obtained. Like all enzymes, ATP synthase can operate in the reverse direction to maintain this equilibrium by hydrolyzing the ATP molecule and pumping the hydrogen ions back across the membrane. And that's chemiosmosis. Thanks for watching.